So we felt that that work really provides a foundation for us to move forward, now knowing that the map position for most of the, or a lot of the SNPs that we're working with. And so we jumped into our tetraploid mapping work, and one population that we're working with that was a key to the SOLCAT project is one that's a cross between Premier russet and Rio Grande russet, and it was a cross made by Rich Novi and in the USDA ARS organization in Idaho. And this population has been grown in 3 locations over 2 years for us to collect phenotypic data, and then we've also done the SNP genotyping here at Michigan State University in our SNP genotyping lab. And so I have to give thanks to Rich Novi and Craig Yencho and Christian Thill for collecting the phenotypic data, which will be part of the QTL analysis. The traits that were collected in this population is shown here, but the top 3, specific gravity, chip color, and glucose and sucrose, were some of the key important traits. And the data was replicated twice, and we used 10 plant plots as our experimental design. Today we're going to focus on, when we're looking at the QTL analysis, on the specific gravity data that was collected. For people that are not familiar with potatoes, specific gravity is an estimate of the potato tuber's density, which is has a good correlation with the starch content of the potatoes. So a higher starch content is desirable for the processing of French fries and potato chips. This russet mapping population is part of a broader set of germplasm that we've looked at in SOLCAT. Also in this broader population is our diploid mapping populations, which I've talked about already, as well as our diversity panel, which I mentioned earlier. And in this diversity panel, we have 325 clones that we've SNP genotyped, and of those 325, we've collected phenotypic data on about 220 of those lines over 2 years at multiple locations in the U.S. So that's going to be part of some future analyses. There's a number of other populations that we've SNP genotyped under the SOLCAT umbrella, and these are listed here. The first 5 populations are tetraploid populations, and the one at the bottom of the list there is a diploid population. And you can see the traits that we're measuring or in these different populations, and it'll be very interesting as we start get through the QTL analyses and being able to validate certain QTLs for similar traits across the different mapping populations. Just for people that are not familiar with the SNP development as part of the SOLCAT project, our SNPs were developed from elite germplasm, so we used 3 varieties, Snowden Atlantic and Premier Russet. And from those lines we collected RNA from 4 different tissue types, as you see here, tuber, leaf, flower, and callus. And from there we were able to then get a good broad look at the transcriptomes of these 3 varieties. We used Illumina GA2 sequencing. Alan Van Dynes did the work at UC Davis and generated approximately 2 gigabases of sequence information on each of the lines, which gave us about 50 to 60x coverage of the transcriptome. Robin Buell's lab, with a lot of work from John Hamilton, did the bioinformatics work and sorted through all the SNPs, and we had over 500,000 different SNPs that we could consider for using. But to fit the Illumina Infinium platform design, we had to go through a certain specific filtering steps. And when it was all done, we had 69,011 SNPs that passed all the filtering steps. And all these SNPs can be found on the Potato Genome browser. So we chose to make a 10K Infinium array for the potato. And when it was all said and done, we had about 8,000, well, we had actually exactly 8,303 SNPs that were in this SNP array for potato. And if we look at what made up these SNPs in this final number, was about 3,000 of them were from candidate genes that were suggested by the community. So we found all the SNPs that we could in those. Plus, we found some SNPs that were in previously mapped genetic markers as a reference point. Plus, the remainder of the SNPs coming from ones chosen throughout the genome to maximize our genome coverage. The Infinium, Illumina Infinium system is based upon single base extension, and so just to kind of walk you through that platform design of our potato array is that what you have is a large number of beads, and on each of these beads is an oligo specific to the SNP that we're trying to identify. And Illumina creates a bead pool of about 250,000 beads per sample. And that sample over here is one of these 24 wells on this chip that you see over here. And then what goes through is a random self-assembly of these beads onto the chip. You can see them going into the wells over here. And then when you average this all out, there's a redundancy built into the system. So for each SNP that we're trying to read, there's about a 15 to 30 bead redundancy of that SNP. And so when we do get a call, it's really based on the average of those calls for that SNP. And so we have the 8,303 SNPs on a chip, and on each chip we can have 24 samples. It's about a 3-day operation to do the SNP analysis if we have good quality DNA to start with on the first day. And when the process is done, the bead chips are loaded into the Illumina iScan, and then a laser is used to excite the red and green fluorophore, and that then is read into the computer software, and it records this high-resolution image, and you can see this over here, and you're getting the red and green emission of the light from the different beads. So this data then is matched up with the mapping software that comes from Genome Studio of where each SNP is on the chip, and then that's imported into our Illumina Genome Studio software.